The overall goal of this procedure is to isolate whole organ biological matrices from the lungs of rhesus macaques and to use these matrices as scaffolds for investigating lung tissue engineering applications. This is accomplished by first decellularizing whole rhesus macaque lungs with detergents, salts, and enzymes. The second step is to install the decellularized lung scaffolds into a bioreactor capable of both ventilating the airway and perfusing the pulmonary vasculature with liquid cell culture media. Next, the lung scaffolds are seeded with stem cells via the airway and endothelial cells via the pulmonary vasculature. The final step is to culture the cells within the lung scaffold under the ventilation and perfusion conditions provided by the bioreactor for the desired amount of time, followed by analysis of the resulting cellular and tissue morphology. Ultimately, recellularization of acellular macaque lung scaffolds with stem cells and endothelial cells results in engineered tissue that mimics the anatomical and histological features of native lung tissues.
This method can help answer key questions in the lung tissue engineering field, such as whether stem or progenitor cells can be used to regenerate pulmonary tissue either alone or in concert with mature pulmonary epithelial and endothelial cells.
The implications of this technique extend toward therapy for end-stage lung diseases because recellularization of acellular lung matrices with patient-derived stem or progenitor cells has the potential to increase the number of donor lungs available for lung transplantation. Moreover, the resulting